In class, we've already introduced the topic of microbiology when we took a look at the history of the science. What we'll do here is go over a few more introductory things about microbiology, and then after that we'll take a look at the structure of bacterial cells. One thing that's useful to know about bacteria is that they come in different shapes and sizes, and they can be categorized according to what shape they come in. Here are three of the major shapes that bacterial cells come in. There are some that have a circular shape, and whenever that's the case, they're described as a coccus shape. There are bacteria that are shaped like a rod, and whenever that's the case, it's said to have a bacillus shape. And another general shape that's pretty common is a spiral-shaped bacteria. And so whenever you have bacteria like this, they're referred to as spirillum bacteria. It turns out that these terms, coccus and bacillus, have plural forms and singular forms. So the way that you're taking a look at the terms right now, these are the singular versions of the word. So whenever you see just one circle-shaped bacterium, you would say that you see a single coccus under the microscope. But if you have a whole bunch of these types of bacteria, the plural for coccus is cocci. So you see one coccus, or you see a whole bunch of cocci underneath the microscope. Same thing goes here. So bacillus, that's the singular form of the word. If you see a whole bunch of rod-shaped bacteria under the microscope, you would say that you see a whole bunch of bacilli under the microscope, because bacilli is the plural version of bacillus. Right, so there's different shapes that bacteria come in, and and there's a singular and a plural version for each one of the shapes. Another way that you can categorize bacteria is according to the arrangements that they form. So when the bacteria divide, some bacteria kind of stay grouped together, at least temporarily, after they divide. And so the way that they group together can be unique depending on the type of bacterium it is. So if you have bacteria that tend to be by themselves, like you see here, they're said to be in a mono arrangement, M-O-N-O, because mono means one or single. If you have bacteria that tend to stay together as a pair, like this, then the technical term for that is a diplo. So diplo refers to the arrangement. These two guys are stuck together as a pair, and then coccus refers to the actual shape of each one of the individual cells. So the arrangement is described here as diplo, diplo meaning two. If you have bacteria that tend to form an arrangement with three other bacteria, so four in total, then that's called a tetrad arrangement. Another common arrangement is to have bacterial cells strung together in a single chain, and so whenever that's the case, that arrangement is referred to as a strepto arrangement. Strepto referring to chain, and again coccus referring to the shape of each one of the individual bacterial cells. And then the last common arrangement that we'll name here is one that involves the bacteria being bunched together like a grape cluster. And so whenever you have that, a grape cluster arrangement, that's referred to as a staphylo arrangement. So staphylo literally means grape cluster, and again coccus refers to the shape of each one of the individual bacterial cells. So one of the things that you're going to be asked to do is to be able to identify shapes and arrangements of bacteria under the microscope. So you're going to be asked to do this in the lab. Here are nice, neat little drawings of each one of the shapes and arrangements, but the shapes and arrangements might look a little bit different under the microscope. So to give you a preview of what we're going to be doing and looking at in the lab, how about if we do this? So here is a picture of some real-life bacteria. Well, they're not alive anymore, but these are real bacteria underneath the microscope. And so you might be able to identify that bacteria that have this particular shape, again, are referred to as bacillus if it's just one, or bacilli if there's a whole bunch of them. And so here's what real-life bacilli look like under the microscope. The circle-shaped bacteria, the coccus, look like this under the microscope. And so you might be able to tell that, well, if you take a look at this picture right here, you might see more than one type of arrangement, right? So you see some that appear to be in a diplo arrangement. You see some that appear to be in a tetrad arrangement. You see a whole bunch more, though, that appear to be in a Staphylo arrangement. So you're going to see this in the lab, too, that you might see many different arrangements for one species of bacteria. So typically what's done is that you take a look at the bacteria, the microscope slide as a whole, and whatever arrangement you see the most of, then that's what arrangement the bacteria is said to have. So because most of these bacteria are arranged in a grape-like cluster, these would be referred to as Staphylococcus. It could be that these smaller groupings here are just bacteria that kind of broke off of one of the bigger Staphylo arrangements. So these are cocci. They're grouped like grape clusters, so they're referred to as Staphylococcus if you're going to talk about the arrangement. One of the things that is a little bit tough to identify as far as shape is if you have bacteria that look like this under the microscope. So you might think that these are cocci because you see some that look kind of circular, but if you look really close, they're not as circle as these real cocci here. So you see how this one, for example, it's pretty much a perfect circle, and so that's what would indicate a coccus shape. These are more kind of ovoid, and it's kind of more obvious here that that's not a perfect circle. So whenever you see this, not quite a perfect circle, kind of ovoid looking, those are classified as bacillus. It's just that they're really short bacillus. So whenever you have rod-shaped bacteria, whenever you have bacilli, it turns out that different types of bacillus can be different lengths, right? So not all the bacilli are going to be really long and obvious like this. Again, there's going to be some that are just shorter, but still bacillus because they're not perfect circles. All right, so again, a kind of a useful thing to know about bacteria is that they come in different shapes. They come in different arrangements. We mentioned that when we were looking at the shape of the individual bacteria that there are singular forms of the word and there are plural forms of the word. The same thing goes for the word bacteria itself. So it turns out that whenever you say the word bacteria, that's the plural form of the word. So bacteria implies that you have a whole bunch of cells that you're looking at. If you only have a single bacterial cell that you're looking at, then the word to use is not bacteria, but instead you'd use bacterium. Right? So bacterium then is the singular version. So you would say that you see a whole bunch of bacteria under the microscope, or if you only see one cell, you would say that I just see a single bacterium under the microscope. And so yet another thing that's pretty useful to know about bacteria is that all of the bacteria that we know of is given a scientific name so that we can identify it. And so what we'll talk about last year as far as introductory stuff is basically how bacteria are named scientifically. It turns out that we might know this from other classes, but all scientific names are made up of two parts. And so to show you an example of what a two-part scientific name looks like, we'll look at my favorite bacterium of all time, which is Neisseria gonorrhea. It's the bacteria that causes gonorrhea. And here's a picture of my favorite bacterium of all time. It turns out that it's a Diplococcus. So you always see them in pairs. And so, again, we're taking a look at Neisseria gonorrhea as an example of how scientific names are written. So, again, scientific names have two parts. The first part here is called the genus name. The second part is called the species name. So this first term here tells you to which genus this bacterium belongs, and then this second part tells you to which species these bacteria belong. And so by convention, the genus name is always written, it's always capitalized, and the species name is always written in lowercase. If you're typing out scientific names, they're always italicized. On the other hand, if you're handwriting a scientific name, it's kind of handwrite in italics. So instead what's done is that you underline it if you're handwriting it. And so, again, for example, if you're handwriting this in a sentence, you would say, Neisseria gonorrhea is my favorite bacterium, and then you would underline it again if you're handwriting it. So this is actually something that I'm going to be looking for in your lab notebook. So every time you write something in your lab notebook, and you have to write the scientific name of a bacterium in there, I'm going to look to see that you're using this proper format, that you're capitalizing the genus, you're leaving the species in lowercase, and then you're underlining the entire scientific name. It turns out that if you take a look at the scientific name of a bacterium, it can actually tell you some stuff about the bacterium. So one common way of naming bacteria is according to who discovered that particular bacteria. So this guy right here, my favorite bacterium of all time, Neisseria gonorrhea, it's called Neisseria because it's named after the person that discovered it. So it turns out that there's a German microbiologist named Albert Neisser that discovered that this is the bacterium that causes gonorrhea, and so it ended up getting named after him. I'm not sure how much of an honor that is to have an STD named after you, but it was done nonetheless. There are other instances where bacteria are not named after the person that discovered them, but instead they're named after what they look like under the microscope. So here's some examples of that. There's a type of bacterium called, its scientific name is Streptococcus pyogenes, and this is one of the main bacteria that causes strep throat. That's actually why strep throat is called strep throat. The strep is short for Streptococcus, which is the genus name of the bacterium that causes that type of infection. And so again, you see here that the genus name actually reflects what these bacteria would look like if you took a look at them under the microscope. Another example of where that's the case is this scientific name right here, Bacillus anthracis. This is the bacterium that causes anthrax. And so you see that the person that decided to give it its genus name decided to have its genus name reflect what shape it is when you look at it under the microscope. All right, so again, bacteria, some general stuff to know about bacteria. They come in different shapes, different arrangements, and then there's a proper convention to use when you're writing their scientific names. So now that we have the introductory stuff out of the way, what we'll do next is take a look at the structure of prokaryotic cells, and mainly we'll focus on bacteria when we're looking at the structure of prokaryotic cells. So in cell biology, you spent a lot of time taking a look at the structure of cells, mostly animal and plant cells though. And again, here what we're going to focus on in microbiology are prokaryotic cells like bacteria and archaea also a little bit. Prokaryotic cells have a different structure than eukaryotic cells, but there's some similarities as well. So that's shown here on this picture. All cells, regardless of whether they're prokaryotic or eukaryotic, they share some commonalities. So here's one commonality that all cells have. They all have a plasma membrane that kind of separates the inside of the cell from the outside of the cell. All cells have cytoplasm, which is the jelly-like stuff that kind of fills up the entire inside of the cell. Remember that cytoplasm is made mostly of water. All cells have DNA as their genetic material. You might remember that in eukaryotic cells, the DNA is surrounded by its own membrane, right? So there's a nucleus in eukaryotic cells. On the other hand, in prokaryotic cells, the DNA is just kind of free-floating, right? It's not surrounded by its own membrane. In other words, you don't see a nucleus inside of prokaryotic cells. And then another thing that's common to all cells, prokaryotic and eukaryotic, is that they all have ribosomes. And you might remember from cell biology that ribosomes are the smaller structures inside of cells where the genetic material goes to in order to tell the cell how to make proteins. So it's at the ribosomes where all of the proteins are produced inside of that cell. So again, here are some things that all cells have, whether they're prokaryotic or eukaryotic. What we'll do for the rest of the time here is focus on the structure of just prokaryotic cells. And so what we'll do here right off of the bat, and actually maybe it's worth mentioning that this is not drawn to scale. So bacteria aren't roughly the same size as eukaryotic cells. Instead, bacteria are typically about 100 times smaller than eukaryotic cells. So this bacterium is just drawn really big so that you can see the structures more clearly. Again, what we'll do is focus on the structure of prokaryotic cells, and right off the bat what we'll tell you is that there are some things that you find only in prokaryotic cells, but not in eukaryotic cells. And so some examples of structures that you find only in prokaryotic cells include a cell wall that's made of peptidoglycan, and we'll talk more about this here in a minute. Capsules are things that you find only in prokaryotes, but not in eukaryotes. Same thing for this stuff called plasmid DNA. You don't find that in eukaryotic cells. And then one more example is are these things called endospores. So these are things that you find again in bacteria, but not in eukaryotic cells. You don't. This picture here doesn't actually show you these four things we just listed. We'll see those in more detail when we look at more detailed pictures later. But we're just giving you kind of a preview here of some things that we're about to talk about. So here's what we'll do. We'll eventually take a look at these structures that are unique to bacteria here in a minute, but before we do that, we'll take a look at and say some stuff about the membrane of prokaryotic cells. So we'll just kind of go through and take a tour of some of the major structural components of bacteria, prokaryotic cells more generally. Again, starting with the membrane. You should have spent quite a bit of time in cell biology taking a look at the structure and the function of cell membranes. So what we'll do here is just quickly review what the general structure of a cell membrane is, and then we'll point out that there are actually differences when you take a look at the structure of prokaryotic cell membranes versus the structure of eukaryotic cell membranes. So here's a picture of what a typical eukaryotic cell membrane looks like. And so you might remember that all cell membranes, well, almost all cell membranes are made mostly of these molecules called phospholipids that are arranged in two layers, or a bilayer. And so in this picture here, all of these orange circles with the two orange squigglies coming off of it, that represents a single phospholipid. You might remember that this circle represents a phosphate head, which has a charge, which means that it's hydrophilic. And these tails represent the fatty acid tails, the lipid portion. And because these fatty acid tails don't have a charge, they don't have a full charge, they don't have any kind of partial charge. Because they lack a charge, they are hydrophobic. And so that's, again, the reason why the phospholipids arrange themselves in this way to form a membrane. The heads are facing areas that typically contain water, like the outside of the cell and the cytoplasm, whereas the tails are snugly kind of hidden from the areas that do contain the water. So again, membranes, they're mostly made of phospholipids. There's some other stuff in there too, like proteins that do things like serve as pores or serve as enzymes. There's some carbohydrates that are kind of sticking out away from the cell. And also in eukaryotic cells, you might remember there are some cholesterol molecules in there that kind of help to stabilize that membrane structure. So again, this is what a typical eukaryotic cell looks like. It's pretty much what all cell membranes look like, in that they're all made mostly of phospholipids. What we'll do is point out one difference in prokaryotic cell membrane. So here's what a membrane of, say, a bacterial cell would look like. Again, it looks very similar. So when you look at it, you might not see any difference at all, right? It's made mostly of phospholipids that have arranged themselves in a bilayer. There's some stuff stuck in that phospholipid bilayer. There's some proteins in there. There are some carbohydrates sticking out away from that from the membrane structure. The one difference we'll point out, though, is this. Remember that in that eukaryotic cell membrane, it contained these molecules called cholesterols that serve to kind of stabilize the cell structure. Bacterial membranes don't have cholesterols, but they have something very similar called hopanoids. So they do a similar thing in that they you find them every now and then in the membrane, and they help stabilize the structure of that membrane. The reason why they can have a similar function is because their structure is pretty similar. So if you're interested in taking a look at the chemical structures, you won't be asked to know them, but if you're a chemistry kind of a person, you'll see that, again, the hopanoids in prokaryotes have a very similar structure to the sterols. And an example of a sterol is cholesterol that you find in eukaryotic cells. So there's several ring structures involved there. So again, we're taking a look at prokaryotic cell structure. Specifically, we're taking a look at the membrane of prokaryotic cells. We're saying that there's some differences in prokaryotic cell membranes that you don't find in eukaryotic cell membranes. One of those differences is this. Bacterial membranes have these things called hoponoids that kind of take the place of the sterols that you find in eukaryotic cells. It turns out that that's not the only difference between prokaryotic cell membranes and eukaryotic cell membranes. Another difference is that there are some bacteria that have a membrane that don't have phospholipids as one of the layers, and instead they have these things called lipopolysaccharides. So we'll list that here. That another difference that you find in prokaryotic cell membranes is that there are, again, some bacterial membranes that have this molecule called lipopolysaccharide, or LPS for short, and they have that in place of phospholipids. So we're mentioning it here now, but we'll show you actually more about this, what lipopolysaccharides are and what lipopolysaccharides do. We'll say more about that a little bit later on, but it's worth mentioning here that this is another difference that you'll find in some bacterial membranes. We mentioned that the basic function of the cell membrane is that it separates the outside of the cell from the inside of the cell. But you know that it is possible for molecules to get across that membrane, right? So it's necessary for a cell to be able to take in nutrients. In order to take in nutrients, it has to have those nutrient molecules somehow get through its membrane. And so what we'll take a look at next is the general types of transport that can occur across the membranes of bacterial cells. So we'll take a look at three general types of transport that can occur. One type of transport is called simple transport. And so again, we're talking about ways here that things can get through the membrane of bacterial cells. Simple transport involves a pore protein that you find kind of spanning that bacterial membrane. And what happens during a simple transport is that the molecule that the cell is interested in taking in, for example, a nutrient molecule, it can only go through the pore if something else goes with it. And typically what goes with it is a proton. And so in order for this simple transport to occur, there has to be a proton gradient present. And so, in other words, there has to be a buildup of protons on one side of the membrane that you don't see on the other side. And so whenever you have this gradient of protons, because there's a lot more here than there are other places, they would tend to want to diffuse towards areas where there are less of them. And so this pore makes it possible, or this pore basically gives those protons a place to diffuse through. And so along with that proton, the molecule that the cell is interested in transporting actually goes with it. And so the energy that's needed to transport this molecule through the cell is actually provided by the proton gradient. When one of those protons goes through, basically takes with it the molecule that the cell is interested in taking in. So a simple transport, to sum that up, it involves a pore protein, and also the energy to transport the molecule is provided by, that should say, diffusion of those protons. Another type of transport that can occur across a bacterial membrane is called group translocation. And group translocation is similar to simple transport in that it also involves a pore protein that kind of spans the entire width of the bacterial membrane. Here's what makes group translocation unique. Basically, the molecule that the cell ends up taking in ends up being modified while it's being taken in. So here's what that looks like. So say that this is a molecule that the cell is interested in taking in. During group translocation, it goes through that protein pore, but in transit, it ends up getting modified. So an example of a modification that commonly occurs during group translocation is the addition of a phosphate group to that molecule. And so the molecule that usually ends up giving up its phosphate so that this molecule can have a phosphate is something called PEP, which stands for phosphoenol pyruvate. You might remember this molecule appearing when you were taking a look at glycolysis in cell biology. But the take-home point here is that this molecule has a phosphate group on it. That's the phosphate group that ends up getting attached to the molecule that's being transported in during group translocation. And so, again, this is what defines group translocation. The molecule that's being transported across ends up being modified while it's being transported. And again, here is an example of a modification that can happen. It gets phosphorylated. In other words, it has a phosphate group added to it. So to sum up, group translocation, it's when transported molecule becomes modified during transit. And one of the advantages to doing group translocation, to having a phosphate group added onto a molecule, is that, well, if this molecule that's being taken in, if that happens to be a glucose molecule, you might remember that one of the ways of breaking down glucose is via something called glycolysis. The very first step in glycolysis involves the addition of a phosphate group to that glucose molecule. So one advantage of a group translocation is basically it gets the process of glycolysis kickstarted, right? That first step of glycolysis can happen while glucose is being transported in. So it just kind of speeds up that process a little bit. Again, what we're taking a look at here are different ways that molecules can get transported across the membrane of bacterial cells. Here are two ways. We'll take a look at one last way or one last example of how molecules can get across a bacterial membrane. And that last example of a membrane transport is called, where it uses something called an ATP binding cassette, or ABC for short, ATP binding cassette. And so this type of transport is similar to the ones that we've just taken a look at, in that it also involves a protein that spans the width of the membrane. What makes it different is that it involves some additional proteins. So it involves something called a binding protein that you find outside of the cell. And it also involves something called an ATP hydrolyzing protein that you find inside of the cell, right? It's associated with the transport protein just inside of the cell. And so basically what happens during this type of transport is that if a molecule shows up just outside the cell, and that cell is interested in taking in that molecule, what happens is that this binding protein basically grabs a hold of that molecule and delivers it through the pore. The way that that molecule ends up being released from the binding protein is that an ATP is used. This ATP hydrolyzing protein is what allows the energy from that ATP to be used to have the binding protein release the molecule. So that was kind of long-winded, so we'll show you what I just said. So if a cell runs across a molecule that it's interested in taking in via this ATP binding cassette, what occurs is that it uses that binding protein to grab a hold of that molecule that it's interested in. That binding protein then delivers it through the pore. In order for that molecule then to get released inside of the cell, this whole thing, that binding protein plus the molecule that the cell is taking in, associates with this ATP hydrolyzing protein. What this ATP hydrolyzing protein does is that it grabs a hold of an ATP molecule and hydrolyzes one of the phosphates off of it, so that you end up getting a phosphate group and an adenosine diphosphate left over. The energy that's released when that ATP is hydrolyzed and is used to change the shape of this purple protein, which then changes the shape of the binding protein, which then allows that molecule to be released to the inside of the cell. So ATP binding cassette involves a few different proteins, and we'll sum that up here. It involves for sure a binding protein, that gray protein that functions to bind the molecule that the cell wants to bring in, and it then carries it through the pore. Once inside, that binding protein again attaches to an ATP hydrolyzing protein, what that ATP hydrolyzing protein does is that it binds to and then hydrolyzes an ATP molecule that then releases the energy that's needed to make the binding protein let go of the molecule that it just brought across. So the transported molecule is then released to the inside of the cell. So again, membranes of bacteria, they have a little bit different structure. And there's different ways that molecules can get through the membrane of prokaryotic cells. So what we'll do next is move away from the membrane and take a look at another part of a bacterial cell, and that part is the cell wall. So to kind of give you an idea of where these things are located, here's a really general drawing of a bacterial cell. What we just took a look at is the membrane of bacterial cells. So in this picture, the membrane would be represented by this kind of dark orange, brownish region of the cell that's surrounding the cell. Again, what we're going to do next is take a look at the cell wall of a bacterial cell. And so where you would find the cell wall is just outside of the membrane. So in this picture here, the cell wall is represented by kind of the darker purple layer in this multi-layered kind of cell. So here are some things about the cell wall. So here's a kind of a little bit better drawing of the cell, of the bacterial cell wall. Again, what we just did was take a look at the membrane. Now we're going to take a look at the cell wall, which is a structure just outside of the membrane. And it's a pretty thick structure too. It's a good thing that it's thick, because the function of the cell wall of bacteria and other cells that have cell walls is that it helps prevent bursting due to osmotic pressures. So bacteria typically find themselves in environments where the liquid in that environment contains a lower concentration of solute than you would find inside the cell. Right? So because the solute concentration is typically higher inside the bacterial cell than it is in its environment, that means that water would tend to want to rush in to the bacterial cell. And so then to prevent that cell from bursting as a result of taking in that water, they have this cell wall that kind of gives it some protection against that. It gives it a kind of a more rigid structure, so it's less prone to bursting as a result of taking in water. The thing that makes up the cell wall of bacteria is this stuff called peptidoglycan. So you might remember from cell biology that other types of cells have cell walls too. Plants have cell walls, fungi have cell walls, algae have cell walls. What makes the cell wall of bacteria different is the stuff that makes it up. So you might remember that plant cell walls are made up of cellulose, which is basically a string of glucoses that are attached to one another in a certain way. The cell wall of fungi are made of this stuff called chitin. The cell wall of bacteria is made up of something else, and it's called peptidoglycan. And so we'll take a closer look at the structure of peptidoglycan here in a second, but we'll mention one more general thing about the bacterial cell wall, and that's that pretty much all bacteria have it. So it's an important structure to prevent, again, that bacterium from bursting. There's only one exception, though. So there is a type of bacterium that does not have a cell wall, and that type of bacteria is something called mycoplasma. And the reason why these bacteria can get by without a cell wall is that if you take a look at, or is because if you take a look at their membrane structure, they have more of those stabilizing hopanoid molecules in them. And they actually, I think, also have sterols in them too. So mycoplasma, again, basically they can get by without a cell wall because their membranes are kind of super reinforced. So here's what we'll take a look at next. We'll, again, focus in on the cell wall. Specifically, we'll take a look at the structure of this peptidoglycan that makes up the cell wall. You might be able to tell from the name of this stuff what molecules you would expect to find in it. You might remember from a different class that whenever you see this term glyco or glycan, that refers to sugars. And whenever you see this term peptide, basically peptides are small proteins, so they're shorter chains of amino acids. And so that's exactly what peptidoglycan is made up. There's sugar chains in there, as well as some short chains of amino acids. and they're arranged in a way that looks something like this. So here in yellow are all of the the chains of sugars that you find in peptidoglycan, and then here now in yellow are the short amino acid chains that you find in peptidoglycan. So it turns out that the type of sugar you find in peptidoglycan, it's not glucose, but it's something that has kind of a similar structure. And actually there's two different types of sugars that you find in peptidoglycan. One type of sugar is called N-acetylmuramic acid, or NAM for short. And the other type of sugar that you find in peptidoglycan is this stuff called N-acetylglucosamine, or NAG for short. And so the sugars arrange themselves alternatively. So they alternate NAM, NAG, NAM, NAG, and that's how they form a single chain. And peptidoglycan is actually made up of a whole bunch of these sugar chains basically stacked on top of one another and attached to one another. One thing we should mention here is that you might notice, again, that peptidoglycan is made up of two different types of sugars. Specifically, it's the NAM sugars, the N-acetylmuramic acids. Those are the specific sugars that have the short peptide chains attached to them. So it's only the NAMs that have the peptides to them. The NAGs don't have a peptide chain attached. It turns out that all of these things are actually connected to one another, and so that's what kind of gives peptidoglycan its rigidity, its rigid structure. All of the sugars in the same chain are attached to one another via a type of link called a beta-1,4 link. Each adjacent chain is attached to one another via bonds that form between these peptides. So this chain here connects to this second chain here as a result of bonds that form between these amino acids and these amino acids, and then on the same token, this second chain here connects to this last chain as a result of bonds that also occur between those peptide chains. So something like this is what ends up connecting the adjacent chains to one another. So here's a general structure of peptidoglycan. Just a couple of fun facts here. So you might know that there are certain body secretions. So things like tears, things like sweat, that contain antimicrobials in them. And so one of the major antimicrobial substances in tears and sweat is this stuff called lysozyme. And the reason why lysozyme is good at being an antimicrobial is because lysozyme actually breaks this beta-1,4 link that connects the sugars together in the cell wall of bacteria. So that's one little fun fact. And then another fun fact that we'll actually say more about later on in the quarter is, there's this antibiotic that people can take called penicillin. And the reason why penicillin is effective at preventing the growth of bacteria is because penicillin actually prevents the formation of these bonds that occur between the peptides in peptidoglycan. So basically penicillin prevents peptidoglycan from completely forming, and so that's what ends up doing the bacteria in. Alright, so again, the cell wall of bacteria is what we're taking a look at here, and peptidoglycan, this stuff is the major component of bacterial cell walls. So how about if we do this? So in case you're a fan of chemistry, so you won't be asked to know this, but in case you're interested, here is the complete chemical structure of peptidoglycan. So here are what the sugars look like that comprise the peptidoglycan sugar chains. We mentioned that N-acetylmuramic acid is the one that has a short string of amino acids attached to it. And so here are the specific amino acids that are attached, and here's how they attach to that N-acetylmuramic acid. So if you're interested, you can pause this video and then just appreciate and Google this. But if you're not interested, then we'll go on here and say more about the cell wall of bacteria. So again, what makes the cell wall of all bacteria similar is that they're all made up of peptidoglycan. But there are differences in how that peptidoglycan can be arranged in different bacteria. So there's two general types of cell walls that you'll find in bacteria, and that's what we'll take a look at next. And so here's how those two types of cell walls are similar. Right? So if is the membrane of the bacterium. We mentioned that pretty much all bacteria have peptidoglycan external to that membrane. There are in some bacteria, though, that peptidoglycan layer is relatively thick, whereas in other bacteria, that layer of peptidoglycan is relatively thin. Not only that, but in these bacteria that have the thin peptidoglycan layer, they actually have an extra membrane that's surrounding it. Right? So they have basically a peptidoglycan sandwich in between two different membranes. And so when we say that there are two slightly different arrangements of cell walls in different bacteria, here are those two types. Whenever you have a cell wall that looks like this in bacteria, that's called a gram-positive cell wall. Whenever you have a cell wall arrangement that looks like this in bacteria, that's called a gram-negative cell wall. This gram-positive and gram-negative, it's called that because of the guy that discovered it. His name is Hans Christian Gram. He was the first guy that kind of made the observation that there are basically two different types of bacteria. And the reason why he was able to observe this is because he was kind of experimenting with different ways of staining bacteria so that they can be looked at under the microscope. And he found that when he did a particular type of stain, all the bacteria he looked at were either one color or another color. So all the bacteria he looked at were either purple or pink. The ones that were purple were called gram-positive, the ones that were pink were called gram-negative. And so it was learned sometime after that the reason why those two bacteria end up being different colors is due to differences in their cell wall structure. And so we'll actually learn how to do this gram stain in the lab. It's probably one of the more useful tools that microbiologists use in the lab when they're trying to identify bacteria. And just really quickly, we'll tell you that the reason why these types of bacteria are called gram-positive is because when you stain them with a particular type of dye, they retain that dye throughout the entire process. These bacteria are called gram-negative, negative because they end up losing that same dye during that staining process. So there's two general types of bacterial cell walls, gram-positives, gram-negatives. What we'll do next is take a closer look at the structure of gram-positive cell walls, and then we'll take a closer look at gram-negative cell walls. We'll see that there are other things that make gram-positives different from gram-negatives. So again, first we'll take a look at gram-positives a little bit more closely. So here's a slightly different drawing of a gram-positive cell wall. You can tell again that it's a gram-positive because it has a relatively thick peptidoglycan layer, and it has no membrane that's external to that. One of the things that you find in gram-positives, besides that really thick peptidoglycan, are these things called teichoic acids. So this is unique to gram-positives. You see here that there are two types of teichoic acids. There are teichoic acids that are relatively short, right? So they extend out away from the bacterial cell, but beyond that, they don't go that far into the bacterial cell. They just kind of stop within the peptidoglycan somewhere. So whenever you have teichoic acid like this, it's referred to as wall teichoic acid because it just kind of stops within the cell wall of the bacterium. The other general type of teichoic acid is this stuff called lipoteichoic acid, and so that's represented by this entire thing right here. And the reason why these are called lipoteichoic acids is because these are teichoic acids that extend all the way through the cell wall and actually touch the lipid bilayer that you find in the membrane. So again, these teichoic acids are another component of gram-positive cell walls. What these teichoic acids do is this. We'll list three functions and characteristics of teichoic acids. One of the things that these things do is that they can bind to magnesium ions, which then allows the magnesium to get taken into the cell. And magnesium ions are important because they are one of the kind of cofactors that's needed in order for DNA polymerase to replicate DNA. So ultimately, this magnesium ion is needed for the bacterial cell to make more copies of its DNA. And then another thing that teichoic acids do, specifically lipoteichoic acids, is that they help anchor the peptidoglycan to the cell's membrane. So it kind of ensures that the peptidoglycan doesn't get lost by the cell, that it doesn't end up floating away. And then a third thing about teichoic acids, it's not really a function, more of a characteristic of teichoic acids, is that they stimulate an immune response. So these teichoic acids here are things that you find just on gram-positive bacteria. The cells that you would expect to find inside of us, so our own cells, they lack teichoic acids. So if our immune system sees any teichoic acids around, then that's a sure sign that there are bacteria around. And so the immune system would want to get activated as a result of that to try to eliminate those bacteria and prevent them from causing an infection. So teichoic acids, these are things you find just in gram-positives. If you take a closer look at, well actually again, if you're a chemistry fan, here is the structure of, the chemical structure of teichoic acid. You won't be asked to know it, but if you're interested, here's what it looks like. If you take a close look at a gram-negative cell wall, you'll see some stuff that's unique to it. So the reason why you can tell this is a gram-negative again is because it has a relatively thin layer of peptidoglycan. Not only that, but it has an outer membrane on top of that. There's some other stuff in here too that's unique to gram-negatives. One of the things that's unique is this stuff called periplasm. And basically what the periplasm is the space between the outer membrane and the inner membrane. And so there are a few things that you would expect to find in this space. So one thing that you can see right here is that you find the peptidoglycan basically sits within that periplasm. But other things can be found in that periplasm too, and so we'll list them here. So things that you find in the periplasm include the peptidoglycan. You also find binding proteins in here. So if you remember when we were talking about transport across the cell membrane, we mentioned that during ABC transport, there's something called a binding protein that grabs onto the molecule and then brings it through the membrane. And so this area here, the periplasm, is where you would expect to find those binding proteins in gram-negatives. Also within this periplasm are hydrolytic enzymes that might get the process of breaking down a nutrient molecule started. So all kinds of things are kind of housed in the periplasm of gram-negatives. Another structure that's found in the gram-negative cell wall is this stuff called lipoprotein, represented by this two-part yellow molecule here. And you might be able to guess that what lipoproteins do is that they help anchor the outer membrane to the peptidoglycan. So it basically ensures that the outer membrane doesn't go floating away. Another structure that you find in gram-negative cell walls is this protein called a porin protein. And so you might guess that what a porin protein does is that it acts as a pore that allows things to get through the outer membrane of the cell. One last thing that we'll mention about gram-negative cell wall structure is the fact that they have these molecules called lipopolysaccharides. And so in this picture, the lipopolysaccharide is represented by this entire thing. So this yellow portion that's embedded within the outer membrane, as well as this long polysaccharide portion that extends out away from the cell. And so it turns out that these lipopolysaccharides are thought to do a few things for the gram-negative cell, and so we'll list them here. One of the things that it does is that it binds to magnesium ions so that the magnesium ions can get transported inside of the cell. And so in this way, lipopolysaccharide that you find in gram-negatives are analogous to the teichoic acids that you find in the gram-positives. They're not only analogous in that way, but also in this way as well. It turns out that lipopolysaccharides, whenever our immune system sees one of these molecules, it should recognize it as a foreign thing and respond to it. So lipopolysaccharide stimulates an immune response. And that makes sense because our own cells, the cells that make up our bodies, don't have lipopolysaccharide on them. So if our immune system sees this molecule, it should recognize it as something foreign, something that's found just on bacteria, and hopefully act against it to get rid of the bacteria. So lipopolysaccharide, you might remember that we actually mentioned lipopolysaccharide a while back. And so if you remember a while back, or before we do that, how about if you mention this? Lipopolysaccharide is actually made up of two parts, like you see here. So this part here of the lipopolysaccharide, this orange portion that's embedded within the membrane, it's called the lipid A portion of lipopolysaccharide. This portion here that extends out away from the cell, that's called the polysaccharide portion of lipopolysaccharide. So it's made up of a whole bunch of sugars, basically. So again, back to lipopolysaccharide in general. So we mentioned lipopolysaccharide before, and if you remember, we mentioned it because we said that there are some bacteria that have a unique looking membrane, and what makes the membrane look unique is that instead of phospholipids, they have a layer of these things called lipopolysaccharides. And so you're seeing that here. So you see, again, the membranes of most cells, right, look like this inner membrane does of this gram-negative cell wall, and that it's made up of, or of this gram-negative bacteria. So you see that this inner membrane is made up of two layers of phospholipids. So most cell membranes look like this. Here's one exception. So it turns out that the outer membrane of gram-negatives are made up of two layers, one of phospholipids, but the second layer is not phospholipids. Instead, it's this stuff called lipopolysaccharide. So we're kind of, again, bringing this idea together with something that we mentioned before. And that's that the outer membrane of gram-negatives has a unique structure. And again, what makes it unique is that it's made up of one layer of phospholipids, and then the other layer is made up of not phospholipids, but lipopolysaccharide instead. And so after taking a look at all of this, and if you're deciding that you're now a huge fan of lipopolysaccharide, feast your eyes on this. So here's the complete chemical structure of lipopolysaccharide. Here is the lipid A part, the part that's embedded within the membrane, and then here is the polysaccharide portion that extends out away from the membrane. So again, you won't be asked to know the complete chemical structure, but in case you're interested, here it is. What we will mention though is this, kind of a preview of something that we'll be talking about later. It turns out that this lipid A portion of lipopolysaccharide, it acts as a toxin. So if you have any bacteria inside of you, and for some reason if this lipid A portion of the bacterium becomes released, and if that lipid A ends up getting released into the blood, that ultimately causes fever and a sudden drop in blood pressure. So it's referred to as endotoxin because of that. So what we'll do next is leave the cell wall behind, and we'll focus on another bacterial structure that's called the capsule or the slime layer. And where you would expect to find the capsule or slime layer is that it's external to the cell wall. So back to our kind of general picture of a bacterial cell, the cell membrane in this picture is the innermost kind of layer, and then outside the cell membrane is the cell wall, which we were just looking at. And for the bacteria that have capsules or slime layers, you would expect to find that outside of the cell wall. So in this picture, the capsule is the light blue thing. That's the outermost kind of covering of this bacterial cell. So we'll take a closer look at the capsule or slime layer of bacteria. Capsules and slime layers are basically the same thing. They're made up of the same stuff, and they do the same things. The difference, though, is basically how neatly organized, how neatly packed together the structure is. So you can see that kind of contradiction in structure here, where you can see the differences in the structure here. So whenever you have a capsule, that's a structure that's more kind of tightly organized around the outside of the bacterial cell. Slime layers, again, they're made up of the same stuff, but they're not as tightly organized around the outside. They're more kind of diffuse and loose. And it turns out that the slime layer, it's easier to separate that from the bacterial cell than it is to separate the capsule from a bacterial cell. So the slime layer and capsule, they're different, again, in that the slime layer is a looser structure that's easier to separate from the bacterial cell. Here's what capsules and slime layers have in common, though. So they both are, again, made up of the same stuff, and typically capsules and slime layers are made up of either polysaccharides or polypeptides. It turns out that if the capsule or slime layer happens to specifically be made up of polysaccharides, then there's a term that's used to describe that, and that term is glycocalyx. So whenever you have a glycocalyx, that's a capsule that's made specifically of polysaccharides. Another thing about capsules and slime layers, again, they're both made up of the same stuff and they both do the same thing. So capsules and slime layers, they function in adhesion or protection. And so examples of things that encapsulated bacteria can stick to include your teeth. So one of the main bacteria that causes cavities, the reason why they can do that is because they have capsules that allow them to stick to your teeth. And whenever they have access to certain types of sugars, one of the byproducts of that sugar metabolism is acids. And those acids can bore a hole in your teeth. So teeth are an example of something that encapsulated bacteria can stick to. There's some bacteria that use a capsule in order to stick to rocks, and particularly if it's a rock that you find on a river. So these bacteria stick to the rock because of their capsules, and they just kind of sit there and allow the river to bring nutrients to them as the river kind of flows over them. So rocks, teeth are examples of things that encapsulated bacteria would stick to. An example of something that a capsule could protect against are our own white blood cells. So it turns out that there's some bacteria that can cause disease as a result of them being able to prevent from being eaten by certain types of white blood cells that usually patrol our blood. So that's an example of something that capsules can protect bacteria against. Turns out that not all bacteria have capsules or slime layers, only some of them do. And then one term that's associated with capsules is this term called biofilm. So biofilm is just a huge mass of bacteria that have capsules that are growing on a surface. So you might find biofilm on your teeth. You might find biofilm on a rock that's in a river. When biofilm forms on medical equipment, that can be a problem. Right? So biofilm is just a whole bunch of encapsulated bacteria growing somewhere. So we'll leave capsules and slime layers behind. They're kind of cool, but we'll leave them behind now and take a look at yet another structure that you might find on bacteria, and that structure is the bacterial pili. And so pili are these relatively short kind of appendages that stick out away from the surface of the bacterial cell. A little bit later on, we'll take a look at flagella, which are also appendages that stick out away from the bacteria. It turns out that pili, though, are quite a bit shorter than flagella. And so again, here's a picture of a couple of bacterial cells that have pili. It turns out that there are different types of pili that a bacterial cell can have, and different types of pili do different things. And so we'll just list the different things that pili can do, and we'll tell you what those different types of pili are called. One thing to mention right off the bat about pili is that not all bacteria have these structures, so only some of them do. Out of the bacteria that do have pili, here's what the pili can do for the bacteria. So there's some pili that function in adhesion. So we just saw that capsules can help bacteria stick to things. Another structure that can help bacteria stick to things are pili. And so if you are talking about a pilus, so pilus is the singular for pili. So if you're talking about a pilus whose specific function is to help a bacterium stick to something, then that pilus is termed, those pili are called fimbriae. So this term fimbriae, again, are pili involved specifically in adhesion. There's other types of pili too, so not all pili are fimbriae. There are other types of pili that function to help reel in other bacteria. And so here's a picture of that. So here's a bacterial cell. There's some bacteria that can shoot a pilus off that actually reaches out and grabs another bacterium in the environment for the purpose of bringing that bacterium in. So it reels it in, right next to it and actually touches it. And we'll see later on that the reason why it does that is so that it can exchange genetic material with it. So whenever you're talking about this type of pilus, one whose purpose is to basically reach out and grab another bacterium and bring it close, that type of pilus is called a sex pilus. It's called a sex pilus again because then after it reels it in and brings it in close, nice and snug, it's able to exchange genetic information with that bacterium. And then a third type of pilus that we'll mention functions in helping bacteria move across certain surfaces. And so whenever you have a pilus whose function is motility across the surface of something, that's called a type 4 pilus. And so here's a picture of a type 4 pilus. So here's bacteria that have, it's really light in this picture, but you have these hair-like extensions running away from the bacteria. That's the type 4 pilus. And basically, the way that these pili help bacteria move across surfaces is that they kind of act as grappling hooks. Right? So the bacteria kind of shoot these out into an area a little bit in front of it. Those pili grab onto the surface, and then it retracts, and that brings the bacteria kind of along with it when it retracts. And so here's kind of a neat little video of real-life bacteria undergoing this thing called twitching motility. So the type of motility that type 4 pili allow bacteria to do, it's called twitching motility. So twitching motility is the type of motility that certain bacteria can do across the surface of things like agar. And so here is video of a type of bacteria called Pseudomonas that is able to do this twitching motility because it has type 4 pili. So here on the left you see a whole bunch of these Pseudomonas bacteria, and you can see that they're moving across the surface of this agar. You see here this boxed-in region. To the right here then is what that boxed-in region looks like up close. So again, you see all of these bacteria just kind of moving across the surface of this agar. All right, so pili, these are, again, things that some bacteria have, and there are different pili that do different things. What we'll do next is take a look at another structure that kind of looks like pili, but not quite, and that structure is flagella. And so here's a picture of a bacterial cell that has actually multiple flagella. Again, flagella are longer than pili. And what flagella do is that they're involved in movement. So unlike the type of movement that type 4 pili do, flagella allow bacteria to move through a fluid. So it allows for movement through fluids, not across surfaces like that, like the pili does, but through fluids. It turns out that not all bacteria have flagella. And it actually turns out that bacterial flagella those are different than the eukaryotic flagella that you might have taken a look at in cell biology. And so what we'll do is point out those differences between bacterial flagella and eukaryotic flagella. So you might remember that an example of a eukaryotic cell that has flagella are sperm cells. And so if you take a look at how the flagella of sperm cells allow those sperm cells to move, it looks something like this. So take a look at here in this black box. We'll show you the movement of mouse sperm. Basically, the flagella whips side to side, right? And it's kind of like a whip. And it's that movement from side to side that's what allows the sperm cell to get propelled through the fluid that it's in. Here's one more look. The eukaryotic flagella moving side to side like a whip. So it turns out that if you consider prokaryotic flagella, they move in a different way in order to propel the cell. So here on the right here you see kind of an artist's rendition of bacterial flagella. This part here, this big part, this is the part of the flagellum that's embedded within the membrane. And so here's the long filament of the flagella that you see extending out away then from the cell. So in contrast to eukaryotic flagella that moves side to side like a whip, bacterial flagella moves in a rotary fashion, kind of like a corkscrew. So kind of like this. And so not only does it move differently than eukaryotic flagella, but we'll see here in a minute that the structure of bacterial flagella is a little bit different from the structure of eukaryotic flagella. So that kind of implies that the flagella in eukaryotes evolved separately from the flagella of bacterial cells. So these two structures kind of arose independently from one another because it was an advantage for bacteria to gain motility, and it was also an advantage for these sperm cells to gain motility, but they gained that ability in completely different environments and situations. So to sum up what we just said here, we're saying that bacterial flagella are different from eukaryotic flagella. Here's one way that they're different. Eukaryotic flagella, like the type that you find on sperm, they move side to side like a whip. On the other hand, if you take a look at the flagella of bacteria, they don't move side to side. Instead, they move in a rotary manner, kind of like a corkscrew. So the motions are different. The structure of eukaryotic flagella and prokaryotic flagella, those also differ. So we'll take a look at the structural differences right now. So you might remember from cell biology that if you consider eukaryotic flagella, basically that flagellum is made up of a bundle of smaller tube molecules called microtubules that are arranged in a certain way. So we'll show that here in this black box. So here's one eukaryotic flagellum. Again, it's made up of smaller bundles of these things called microtubules. Oops. So eukaryotic flagellum is made up of a bundle of these microtubules that are arranged in a specific way. So there's 9 pairs of these microtubules kind of around the outside, and then there's a lone pair on the inside. You might remember that this is referred to as a 9+2 arrangement of microtubules, because there's 1, 2, 3, 4, 5, 6, 7, 8, 9 pairs of microtubules around the outside, and then there's 2 microtubules on the inside. So here's what, again, eukaryotic flagella look like if you're to take a cross-section of it. It's a bundle of microtubules. On the other hand, if you take a look at the structure of prokaryotic flagella, it's not arranged like that. So it's basically just more of a solid stack of polymerized proteins. Right, so here's kind of a side view of bacterial flagella, and then here's kind of a cross-section. So you see that it's not organized like a 9+2 structure. It's a little bit different structure. So let's mention that as another difference between eukaryotic and bacterial flagella. If you consider the structure of eukaryotic flagella, it's made up of microtubule bundles that are arranged in this 9+2 arrangement. If you consider the structure of prokaryotic flagella, it has, well, it does not have that same structure. If you take kind of a more general look at the structure of bacterial flagella, you'll see that it looks like this. Right, so here is, you might be able to tell that this is a gram-negative bacterium that we're looking at because it has 2 membranes, an inner membrane and an outer membrane, and it has the peptidoglycan in between the 2 membranes. We're showing you here that the flagella of bacteria is made up of a couple of different parts. So there's a part that's kind of embedded within the membrane and the cell wall of the cell, and then there's the long part that extends out away. So all of these parts here are given names, so we'll identify those next. This part here that is embedded within the membrane and the cell wall of the bacteria, so this whole apparatus here is called the basal body of the flagellum. This part here that is the kind of the first part to extend out away from the bacterium, that's called the hook. And then the long part that does all the where you see all the movement, that's the filament of the flagellum. We mentioned that the flagella of bacteria turn in a rotary manner, like a corkscrew, and so the energy that's needed in order to turn the flagellum in that way is provided by the diffusion of protons, it turns out. And so there are proteins that are associated with the basal body through which those protons diffuse through. And so those proteins, represented by these red things here, are called MOT proteins. MOT is short for motor, so these are called motor proteins. And again, what these motor proteins do is that they allow, so if there's a whole bunch of protons that has been pumped in this area of the membrane, those protons then end up diffusing through these MOT proteins. That then causes this basal body to turn, which then causes the filament to turn. Right, so to sum this up, these MOT proteins, what they do is that they allow protons to diffuse through them, and then that causes the this basal body to end up rotating. And so if you could see what all of this looks like in action, it would look something like this. Right, so you have, again, the basal body, hook, filament. Here is where the protons are going to diffuse through these MOT proteins. When those protons diffuse through, it causes spinning of this basal body, which then causes spinning of the filament, and that's what allows the bacterium to get propelled through the fluid. What we'll show you next is that it turns out that the spinning action that we see can happen in one of two directions. So it can either happen clockwise, like we're seeing here, but then the bacteria can switch things up so that it ends up spinning counterclockwise as well. And so we'll see what the reason for that is during class, but we'll just mention here that the bacterial flagella can spin two different directions. And so there's some proteins that are involved in causing that switch to happen. So right now it's spinning clockwise, but there are things that can make it stop and then start spinning the other way as well. And so depending on whether the flagellum is spinning clockwise or counterclockwise, those will produce two different types of movements in the bacterial cell. And again, we'll talk more about that in class. So the bacterial flagella, it's different from eukaryotic flagella in its structure and the way that it moves. All right, so the next structure that we'll take a look at is bacterial DNA. Again, DNA, this is an important molecule that you actually find in all cells, not just bacterial cells, but all cells have DNA. But it turns out that the DNA in bacteria is a little bit different than the DNA that you find in prokaryotes, for example. And so we'll point some of those differences out right now. So this picture here shows you actually two different types of DNA that you can find in bacterial cells. There's a type of DNA called chromosomal DNA, and there's a type of DNA that some bacteria have that's called plasmid DNA. One of the things that makes bacterial DNA different from eukaryotic DNA is that bacterial DNA is actually an enclosed loop. So bacterial DNA is circular. The DNA that you find in eukaryotic cells, if you take a look at our DNA, it's not circular, it's a linear molecule. So again, some things about bacterial DNA that makes it different from eukaryotic DNA is that it's circular. And not only that, but the DNA in real life, if you take a look at it inside of a bacterial cell, it's not a neat little circle like this, but it's actually folded and twisted and compacted. So it's not only can it be just folded and twisted and compacted, but it can be super folded and twisted and compacted. And so whenever that's the case, it's considered to be supercoiled DNA. And so this supercoiling actually is not unique to bacteria. The DNA in our cells is actually also twisted and folded and compacted, but because the DNA in our cells is not circular, this thing called supercoiling only occurs with bacterial DNA. And so to give you an idea of what supercoiling is and what it might look like, check this out. So again, the DNA in bacteria is unique because it's circular, but it's actually twisted. And so this will show it to you really quick here. It's actually really twisted, and if you twist it a lot, it becomes really compact. And so we'll show you that process a little bit slower now. So here's the bacterial DNA that's circular. These twists can be introduced to it. The more twists are introduced, the more jumbled and twisted it becomes. So if you take a look at the DNA inside of a bacterial cell, it's not really a perfect circle. Instead, it's a circle that's been kind of jumbled up because of all of these twists that have been introduced to it. And it's actually a useful thing to have the DNA all jumbled up, because otherwise it probably wouldn't fit inside of a bacterial cell. All right, so again, bacterial DNA, it's circular. It's not circular in our cells though, and it's often supercoiled. Again, there's, in this picture at least, you see two different kinds of bacterial DNA. There's this stuff called chromosomal DNA, and there's this stuff called plasmid DNA. So we'll talk about the differences between those two types of DNA next. So it turns out that out of these two types of DNA, there's only one that you would expect to find in every single bacterial cell, and that's the chromosomal DNA. So all prokaryotes have chromosomal DNA, and they have just one piece of chromosomal DNA. So again, this is another thing that kind of makes bacteria different from eukaryotes. You might remember that if you take a look at most of our cells have 46 chromosomes in them. In contrast, bacteria, they only have one chromosome. They only have one piece of chromosomal DNA. The reason why you would expect to find all cells to have chromosomal DNA is because if you take a look at the genes found on chromosomal DNA, those are all of the genes that that bacterial cell needs in order to survive in its natural environment. Right? So these are all of the essential genes that are necessary for survival. That's why all bacteria have chromosomal DNA. In addition to that chromosomal DNA, some bacteria have an extra piece, basically, of DNA called plasmid DNA. So again, this is something that makes bacteria unique. We don't have these extra smaller bits of DNA called plasmid DNA. We just have all chromosomal DNA inside of our cells. And so here's what makes plasmid DNA different, the plasmid DNA of bacteria different from the chromosomal DNA of bacteria. One of the things is that, again, plasmid DNA is something that only some bacteria have. Like you see in the picture, plasmid DNA is smaller than chromosomal DNA. And the reason why a lot of bacteria can get by without plasmid DNA is because if you consider the genes that are found on plasmid DNA, they're genes that really aren't needed in order for that cell to survive in its normal environment, but they might provide some advantage in certain situations. So plasmid DNA contains genes that aren't really necessary, but they might provide an advantage. So for example, a gene that would allow this bacterium to be resistant to an antibiotic. Right? So that's really not a gene that's necessary for survival in its normal environment. But if there happens to be antibiotics that make its way into the neighborhood, that would provide an advantage to this bacterium, because this bacterium would be able to survive the effects of that antibiotic. So antibiotic resistance genes are oftentimes found on plasmid DNA. Other things about plasmid DNA is that not all cells have it, but out of those that do, there can actually be multiple pieces of plasmid DNA inside of a single cell. And so there can be one or more than one plasmid DNA inside of a cell. And then one last thing that we'll mention about plasmid DNA is that it's possible for it to be transferred to other bacteria. And so we'll say more about that later when we take a look at bacterial genetics. So again, all cells have DNA, but there are some things about bacterial DNA that makes it different from the DNA of eukaryotic cells. And actually, before we leave bacterial DNA, we'll mention a couple of terms that are often associated with bacterial DNA. So here's another picture of a typical bacterial cell. Remember that bacteria, they don't have an extra membrane surrounding their DNA. The DNA is just kind of floating naked in the cytoplasm. But there's a name given to the region of the cell where the bacterial DNA usually is found. And so that region is called the nucleoid region. So in words, nucleoid, and again, we're taking a look at some miscellaneous terms that have to do with bacterial DNA. One term is nucleoid, and that's just the area of the cell where the chromosomal DNA is found inside of that bacterium. Another term that's associated with not just bacterial DNA, but all DNA, is this term genome. And so whenever you're talking about an organism's genome, it's just its entire collection of genes. So the Human Genome Project, that was when the entire collection of human genes were sequenced. And we're kind of sifting through that right now, actually. So again, bacterial DNA, it's unique from eukaryotic DNA, and here's some terms that are associated with it. Another structure that you find in not just bacterial cells, but all cells are ribosomes. And again, ribosomes are the structures within the cell where proteins are produced. We won't say much about ribosomes here, but we'll just mention that, again, the function is to produce proteins. And we'll also mention that if you consider prokaryotic ribosomes, they have a slightly different structure from eukaryotic ribosomes. And so we'll come back to this later when we talk about antimicrobial drugs. There's some drugs out there that take advantage of this difference between prokaryotic ribosomes and eukaryotic ribosomes. The next structure that we'll take a look at is a structure that is pretty cool because it allows bacteria to survive some really, really harsh conditions. So there are some bacteria. So for example, here's a bacillus that when they sense that there are certain stresses in the environment, so say that it gets really hot, really dry, or if there are certain toxic chemicals in the environment, what these bacteria can do before they die as a result of these environmental stresses, they actually produce a little baby bacterium that's designed to be able to withstand those stresses. And so that's what this thing here represents. So the idea is that when that bacterial cell dies as a result of the stress in its environment, that little baby bacterium ends up getting released. And because of the way that this baby bacterium is built, it's able to withstand the stress in the environment. For example, the UV or the toxic chemicals or the heat. When that stress ends up going away, that baby bacterial cell can sense that, and it can shed all of its protective layers and become a metabolically active bacterium again. And so what that structure is called, this thing that is resistant to really harsh conditions, that's called an endospore. And again, what an endospore is, it's the structure that's formed by some bacteria in response to stress. And it turns out that the only bacteria that can produce these things, these endospores, are bacteria that naturally live in the soil. A little bit more about endospores if you're describing it. Again, it's a metabolically inactive bacterial cell that can withstand these stresses, partially because it's surrounded by all of these extra protective layers or proteins. Some examples of stresses that endospores can withstand. We mentioned extreme heat, so temperatures up to 150 degrees Celsius, which is equal to 302 Fahrenheit. That's pretty hardcore. Endospores can resist drying, and they can also resist tons of a certain amount of UV radiation. Some terms that are associated with these really hardy structures called endospores. There's this term sporulation, and whenever someone says sporulation, that's just the process of making an endospore. So whenever these certain types of bacteria feel stressed, they undergo sporulation in order to produce an endospore. Another term that's associated with endospores is this term germination. So germination occurs when that endospore senses that the stress is gone and it comes back to life, basically. So it's when an endospore wakes up to become a metabolically active cell. And this last term that we'll mention that's associated with endospores is this term vegetative cell. So it's kind of, it might seem kind of backwards, because if you think of maybe a person that's described to be in a vegetative state, right? They're not very active, but the opposite is true. So when you're talking about a vegetative cell with respect to bacteria and endospores, vegetative cell is the metabolically active version of that cell. So whenever an endospore germinates, what ends up being produced is a vegetative cell. In other words, a cell that is once again able to grow and divide and produce other cells. What we'll take a look at next is basically what allows an endospore to survive the harsh conditions. So what do you find in and around an endospore that allows it to do its function? Again, basically what an endospore is, it's It's a baby bacterial cell that's just super reinforced. There are things that you find inside of an endospore that you don't find, though, in metabolically active cells. So if this represents the bacteria inside of an endospore, and here's the bacterium's DNA, one of the things that you find inside this cell are these molecules called dipoclinic acid calcium complexes. And what these things do are two things. So I have it so that these dipoclinic acid calcium complexes are bound to the DNA, and that's because it helps stabilize that DNA during the harsh conditions. Not only that, but they also help to dehydrate the cell. And so dehydrating the cell during tough situations is useful because, one, dehydrating the cell slows down enzymatic activity inside of that cell. So there's a lot of enzymes that need water, basically, in order to work. And so if those enzymes aren't working at full capacity, then it means that the metabolism ends up getting slowed down inside of that cell. And so that's useful if you're trying to sit out a long period of being in a tough situation. So that's one reason why lowering the water content is useful. Another reason why it's useful to lower the content of water inside of a cell that's part of an endospore is because it makes it more difficult for it to get really hot inside of that cell. And so again, remember, one of the reasons why a cell might make an endospore is if it's really dry, really hot. If the cell is somehow able to lower the water content inside of itself, inside of that cell that's in the endospore, basically water, if you remember from cell biology, maybe chemistry, it's really good at retaining heat, right? So if there was a lot of water inside of that cell, if the outside environment was hot and that heat then got inside of that cell, it would stay hot inside of that cell for a really long time, which would end up killing the cell ultimately. So by kind of lowering the amount of water that's inside the cell, it just makes it more difficult for the inside of the cell to get warmed up. And so that's a useful property to have if you're going to be sitting for a long time in a really hot environment. Another thing that you would expect to find inside of a cell, if it's inside of an endospore or if it's part of an endospore, are these molecules called small acid soluble proteins. And so I have those proteins, or those molecules, also attached to the DNA because it also helps to kind of stabilize the DNA during the harsh times. But another function of these small acid soluble proteins is that they serve as a nutrient source. So when the cell starts undergoing germination, here is what those germinating cells use as their nutrient source to help them get through that germination process. Another structure that you expect to find on endospores, so not only all this stuff inside, but again, endospores have these all of these protective protein layers outside that you don't find in a normal metabolically active cell. Not only that, but again, these endospores have to have a way of being able to detect when the environment has become hospitable again, right? So when all the stresses, when the bad stuff has gone away from the environment, it has to have a way of being able to sense that so that it can become a metabolically active cell again. And so the way that it's able to do that is that it has actually receptors on the outside of those protein coats. And what these receptors do is that they are kind of detectors for molecules that would indicate that the environment is good once again. So let's describe these receptors. They're called receptors for germination triggers. And again, things that would cause this cell to germinate are molecules that would indicate that times are once again good. And so examples of molecules that would indicate good times would be amino acids and sugars, right? Because these are nutrient molecules. So if these are once again floating around in the environment, then it would be a good time then to wake up for this endospore so we can start eating all of those things. All right, so again, whenever these things are available, they end up binding to these germination triggers, and that binding allows that, or basically tells this endospore to germinate. And again, if you consider this entire thing, this is what an endospore is, right? So whenever a cell undergoes stresses, and it makes an endospore. That's what that entire endospore looks like. It's this whole thing. Here's a real-life picture of an endospore. So again, it's surrounded by all these protein coats. During lab, we're actually going to take a look at endospores under the microscope. There's a thing about endospores, though. So because of all of these extra tough protein coats that surround an endospore, it's relatively difficult to get stain to go inside of an endospore. So oftentimes, if you see bacteria under the microscope that make endospores, you'll see this clear area inside of it. So the stain was able to color everything else in the bacterial cell, but because of the extra protein layers surrounding the spore, the stain has a really hard time getting in, so it just remains clear like this. So if you see bacteria under the microscope that have like a distinct clear area inside of it somewhere, that clear area is an endospore. We're actually going to do a special kind of stain that is able to penetrate the spore and color it green. So we're going to do a stain that colors the bacteria pink, and any endospores that they contain, those will be colored green. So in a couple of weeks, if you end up doing an endospore stain that looks something like this, that's textbook. That's what it's supposed to look like. You might be wondering, well, you know that endospores, they're these really tough structures that can withstand really harsh conditions. You might be wondering, well, how long can an endospore sit and still be able to germinate? So decades for sure. So people have for sure germinated endospores that have been around for decades. There was a group back in 1995 that reported discovering and then germinating spores that were about 25 to 40 million years old, they estimate. And so where they found these spores were in the abdomen of a wasp that got trapped in some amber. So it got trapped in some sap 25 to 40 million years ago. So when they discovered that this wasp contained endospores, they did what any probably curious person would do, and they tried to bring them back to life, and it turned out that they were able to bring them back to life. There's some controversy here, though, because there's some people that think that the spores that these scientists were able to bring back to life were actually spores from today that just ended up contaminating the sample. So there's some scientists that say, well, wait a minute, you didn't really bring 40 million year old spores back to life. What you actually brought back to life are spores that were just made maybe yesterday, that just made their way inside of your sample. Nobody's really been able to definitively prove that it was a contaminant, so I guess as of now, these scientists are still saying that 25 to 40 million year old spores were able to be brought back to life. So that's pretty impressive. All right, so again, endospores are really cool because they're these structures that allow bacteria to survive really harsh conditions. So for example, they allow bacteria to survive some really hot conditions. What might be even cooler is the fact that there are some cells out there that can survive really harsh conditions even without producing an endospore. So that's even more hardcore. And so it turns out that there's organisms that can live in really hot environments, kind of like this. So these are underwater vents or hot springs. There are types of organisms that not only survive in these types of environments, but they thrive in these super hot environments. In other words, they can only live if the environment is this hot. So the types of temperatures we're talking about here, so there's some microbes that can thrive in temperatures as high as 121 degrees Celsius, which is 250 degrees Fahrenheit. And again, these are cells, the cells we're talking about here are cells that can do this just naturally. So they don't have to produce endospores to do this. And so the type of organisms that are kind of famous for being able to withstand these really hot temperatures, for being able to live in these really extreme environments, are organisms that belong to the domain Archaea. And so remember that when you're talking about prokaryotes, there's basically two types of prokaryotes. There are bacteria and there are archaea. It kind of makes sense that these organisms, these archaea, live in really hot, extreme environments like this because it's thought that archaea were the very first cells to appear on this planet. And in fact, that's why these types of organisms are called archaea. Archaea means ancient, right? So archaea sounds like archaic. So the root of all that is ancient. And again, the reason why these types of organisms are called ancient is because they're thought to have been the very first types of living things to appear on this planet. It makes sense then that these archaea today live in really hot extreme environments, because if you think about the Earth 4 billion years ago, basically the entire planet looked like this. So the reason why these archaea live in really hot environments is because they evolved in an environment just like this, the Earth 4 billion years ago, when the entire planet basically was a hot cesspool of boiling liquid. And so what we'll take a look at next is what allows, what structural features of archaea allow them to live, to thrive in these really hot extreme environments. And so again, if you consider a cell, there are some major molecules that you'd expect to find in all cells, including archaea, right? So all cells have proteins, they all have DNA, they all have some sort of membrane structure. So what we'll take a look at here is what structural components of those molecules allow these cells to thrive in the really hot environments. So first, we'll take a look at the proteins of archaea, and it turns out that the proteins of archaea are a lot more heat stable compared to the proteins of other types of cells. And so when we say that the proteins are heat stable, we mean that it's really difficult for hot temperatures to unfold the proteins that you find in archaea. So if you remember, proteins are just basically a bunch of amino acids that are strung together, and then that big long chain of amino acids is folded in a different way. So different proteins are made up of different amino acids, which means that different proteins fold in different ways to form different shapes. And so that's why different proteins can do different things, because they all have different shapes. You might remember from cell biology that the thing that determines how a protein folds is the sequence of amino acids. And so here we're showing just a general diagram of a protein. So it's just a string of amino acids. You might remember that there are 20 major amino acids out there that can be used to make most proteins. What makes the 20 amino acids different from one another is that they all have a different R group. So I have the R groups just drawn in as these lollipop-looking structures in this picture. And so you might remember that there are some amino acids that have R groups that have no charge. There are some that have a full positive charge. There's some that have a full negative charge. And there's other amino acids whose R groups contain a partial negative charge. You might remember that anything with a charge, including these things with a partial negative charge, those are all hydrophilic, right? Because if it has a charge, then it would be able to bind to one of the partial charges that you would find on a water molecule. If something has no charge, like these guys, then it would be hydrophobic, right? Because it has no charge, it has no way of grabbing onto a water molecule. You know that pluses and minuses attract one another. So those are all the factors that would cause this particular string of amino acids to fold in a particular way, so that the hydrophobic R groups are hidden away from the water, the hydrophilic R groups are exposed to the water, and maybe these pluses and minuses can get together, kind of like this. Right. So again, the shape of a protein is determined by the chemical properties of the amino acids that make up that protein. One of the reasons why hot temperature doesn't easily unfold the proteins that you find in archaea is because if you consider the amino acids whose R groups are found in the core of archaea proteins, those amino acids have really extra hydrophobic R groups that will represent by these yellow things. So because the core amino acids that you find in archaea proteins are very hydrophobic, that basically I see it as counteracting the effects that the heat might have when it starts unraveling or denaturing that protein. So if this is in a really hot environment, this is how I kind of picture things happening, and that heat starts making the molecules move faster that comprise this protein so that it might start unraveling. Because the core amino acids of archaea proteins are very hydrophobic, they are very, very resistant to being in contact with the water. So it'll kind of curl that protein back up into its kind of natural state. So archaea, one of the things that makes them able to survive really hot temperatures is the fact that they have proteins that are heat stable. And one of the things that makes proteins in archaea heat stable is the fact that if you take a look at the amino acids in the core of those proteins, they're very highly hydrophobic. So for example, phenylalanine, the amino acid phenylalanine is one of the more hydrophobic amino acids out there. So you might expect to find a lot more of that particular amino acid in the core of the proteins that you find inside of archaea. Another thing that allows archaea to survive in the really hot temperatures and really hot environments is the fact that it has heat-stable DNA. And so I'll mention two things that makes the DNA of archaea heat-stable. If you take a look at the DNA of archaea, so here's a general picture of DNA. Remember that DNA is made up of two strands. Each strand has in its backbone a bunch of phosphates and sugars, and then in the middle here are the nitrogenous bases. So it turns out that the DNA of archaea have its sugar phosphate backbone reinforced by things that you don't find in the DNA of other types of cells. So for example, there's this chemical called potassium cyclic 2,3-diphosphoglycerate that you find in archaea DNA, but not really in other types of DNA. And the purpose of this chemical is that it helps to stabilize the covalent bonds that you find between the sugars and the phosphates in the DNA of that archaea. Another thing that helps to stabilize the DNA of archaea in really hot temperatures is the fact that it compacts. So actually this isn't unique to archaea DNA. Other cells have DNA that becomes compact as well. But here it's especially important to have that compact DNA because it helps prevent that DNA from denaturing while it's in the really hot environment. So the DNA of archaea, like other cells, is compacted kind of like this. So it's not just a straight up ladder structure. Instead, it's been folded so that it looks more compact inside of that cell. So we'll jot that down here. It turns out that there's proteins that aid in the compaction of that DNA. So another thing that helps stabilize the DNA of archaea in the really hot temperatures is that there's proteins that bind to that DNA. Again, and that helps compact the DNA so it makes it less likely to come apart in the hot temperature. One last thing that allows archaea to withstand really hot temperatures is the fact that it has heat-stable membranes. And so, like we mentioned before, if you consider the membranes of most cells, they're made up of a phospholipid bilayer. It turns out that the major thing that's holding together the phospholipid bilayer is just the fact that the tails of these phospholipids are all hydrophobic and the heads are all hydrophilic. So that's what causes them to kind of flip, that's what causes the tails to flip inwards to be out of contact with the water, and that's what causes the heads to be in this position so that they're in contact with the water in the cell's environment or the water in the cytoplasm of the cell. So that's a relatively loose arrangement, right? So there really aren't a lot of physical connections between this layer here and this layer down here. And so it turns out that for the most part, it's a good thing to have kind of a loosey-goosey membrane because that allows things to diffuse laterally through the membrane, so things like enzymes can diffuse through it. And it also allows things to go in and out of that membrane a little bit easier via simple diffusion. So again, for the most part, it's good to have this relatively loose membrane structure. On the other hand, though, if you're a type of cell that's going to live in a really hot environment, it would suck to have this loosey-goosey structure, because if things get hot, the heat starts making molecules move faster. And if it's really hot, it'll make molecules move really fast, which means that the molecules that comprise this membrane would end up basically getting ripped apart in any temperature that's even moderately hot. So what we're trying to say here is that if you are going to be a cell like Archaea that lives in really hot environments, your membrane probably is not going to look like this. There's going to be a lot more reinforcement that's required in order to live in the really hot environment. And so here's what Archaea membranes look like. Instead of being made up of a bilayer of phospholipids, the molecules that make up Archaea membranes look like this. So it's also a type of lipid, but the two heads are actually covalently and physically connected to one another. And so there's a whole bunch of these molecules then that comprise the membrane of Archaea. And again, if you contrast that with the phospholipid bilayer that makes up the membranes of most other cells, you'll see that this is a lot more difficult to rip apart in hot temperatures as compared to this, again, because there's actual physical connections between the head regions of the molecules. So again, this is a general schematic of what an Archaea membrane looks like. And again, one of the things that characterizes it is that it's made up of lipids that have covalently linked head regions. In contrast, this is what the membranes of most bacteria and eukaryotic cells look like. And again, it's characterized by being made of a phospholipid bilayer, and the two layers of phospholipids aren't really covalently linked to one another. Another difference between the membrane of Archaea and the membranes of bacteria and eukaryotic cells appears in the tail region. So if you take a really close look at the tail regions of the lipids in these types of membranes and you compare that to the tail regions, which are the lipids found in these types of membranes, you'll see a difference. So you might remember that the fatty acid tails here in phospholipids are basically a long string of carbons that have hydrogens attached to them, like this. The tail regions of Archaea are a little bit different. So they're similar in that they're made up of carbons and hydrogens, but it turns out that there tends to be more double bonding that occurs between the carbons that make up the backbone of the tail molecules. Not only that, but there's carbon branches. So for example, here you see that there's, so here's the carbon backbone, but then off to the side there's another carbon-to-carbon bond. You don't really see that in the fatty acid tails of bacteria and eukaryotic membranes. There aren't any carbon offshoots. It's only hydrogens that you see coming out of the sides here. Right? So two more differences then between these two membrane structures is that in Archaea, there are the hydrophobic side chains have double bonds and branches. Actually, that's not side chains, so this should say the hydrophobic tails. These regions here have double bonds between the carbon atoms, and then there's carbon branches that shoot off to the side. Whereas if you take a look at the tails of this type of membrane, you don't see that. Right, so again, Archaea, they're pretty hardcore because they're these organisms that can live in really hot temperatures even without producing endospores. And we just talked about 3 reasons why they can do this. They have heat-stable membranes, they have heat-stable DNA, and they have heat-stable proteins.